Bastard. And now I'm here to talk about some of the work we've been doing in the lab, focusing on the olfactory bulb. But before I say anything of what we are doing, I should say who has been doing the work. If ever I'm doing a talk, say I'm actually lying, because I don't do much science anymore when it comes to hands-on. I mostly come up with ideas and try to get funding. And most of our funding is coming from the Wallenberg Foundation, which is basically the same size as the Wellcome Fund in the UK. And then we have the European Research Council, give a lot of money to our lab, and the Swedish NIH, you can say. And the work today has mostly been done by this guy here, Trans Nordén, who is a PhD student, Anne Winter, a PhD student, and Irene Zanetti, who is a research engineer. In addition, a lot of the work in the beginning was done by a former PhD student in the lab, Besadi Ravani, who now, I should update this, he's not at Stanford University, he's in some kind of like startup company, making double my salary, so he's well off. And then Don and Leslie is involved in some aspect of this work. Okay, as you get older in science, you start thinking about the big questions. Many people go to consciousness, but I went to the question, why do we have a sense of smell? What's the point? Why does every living organism have the ability to detect chemicals? Plants, insects, humans, all different animals, even bacteria, right, can detect chemicals. What's the point of that, right? And my thinking, or my argument, is that the sense of smell has two main functions. It's to tell us when to approach or when to avoid something. And I think the core aspect of this is actually to avoid, right? It's better to run away one time too many than not. And there's a couple of evidence for this, I think. Actually, there's tons of it, but I'm listing just a few. There's a close link between odors and pain. Tend to be the general rule: if something is bad for you, it stings in the nose. If something is good for you, it stings less, right? Humans are very good in classifying odors, but we are extremely bad at identifying odors, because it matters if you can eat it or not. It doesn't matter if it's not orange or clementine, right? We have a very rapid neuroplasticity. If you associate an odor with electric shock, for example, you get changes in most of the structural part of the olfactory system. But you also have this very fast behavioral plasticity. One of the last studies I did here at Monell, we paired an odor with an electric shock, and we can show that people can become up to 220% more sensitive to that specific odorant, but not with enantiomers, just by pairing it with a shock 10 times. So a very fast behavioral response to negative associations to odors. But most of this evidence are coming from some kind of association or learning, right? Which is not a very good principle to have if we would like to have some kind of like avoidance response, right? So learning cannot be the sole mechanism. And I argue that what we should look at is pleasantness or valence, right? And why do I think that? Well, because pleasantness or valence are strongly linked to approach avoidance behavior in our other sensory systems. Inhibition and audition is an anatomically early process in the processing stream. It's also a response to low-level stimulus features. So you don't have to have this evolved representation of what you're seeing or hearing to knowing if you should at least run away from it, right? If someone is screaming, you know instinctively this is probably something I should avoid, right? But also when it comes to olfaction, we know that from the lab of Noam Sobol, he has multiple studies and been replicated by others, that one of the primary dimensions in olfaction is pleasantness or valence. And axis 2 is often labeled as toxicity, or like how bad the odor might be for you, right? But also a study that we did a few years ago now that Joel Mainland here was involved in. We collected pleasantness data from tons of different parts of the world, mostly in indigenous people living in isolation or the sort, but also people in New York or in Bangkok. And what we found was that if you don't assign a label to the odor, you just take chemicals and you have people rank them, you can see that only 6% are actually explained by cultural differences. The other part is evenly split between the individual variants or the odorant itself, right? And a few months later, I was involved in a big project where we tested kids around the world, and we found very similar stuff. When it comes to how kids ranked odors, only 5% could be explained by the culture they came from. So actually, valence is culture independent, even though we believe that is so strongly linked to culture. But that's because the odor object valence is very culture dependent. So what's the requirement then for an approach avoidance signal? Well, it has to be rapid, right? So it has to be processed at an early stage of the processing stream. If it takes a couple of minutes, it's often too late, right? It's very beneficial to be connected to the motor system. And you should have this strong influence of past experience. You need to have a very strong updating mechanisms because you have to learn if something is bad for you, right? Or good for you, right? And what we thought was that olfactory bulb might be an ideal point for this, right? The very first synapse from the olfactory receptor neurons. So we hypothesized olfactory bulb process odor valence, and which then will support, of course, the notion that olfactory bulb is involved in approach avoidance behavior. The problem we had when we started this project was that we couldn't find any method to actually record from the olfactory bulb. So almost everything we know from the olfactory bulb is derived in animals, and 99% of that in rodents, right? Because most of the method we use in humans, we cannot get any good signals from the olfactory bulb. And we know that in rodents, the olfactory bulb is involved in pleasantness of odors. There's a couple of studies linking olfactory bulb processing with the valence of the odor. The problem, though, with those studies is that is it really valence or is it pleasantness? We don't know, right? Because what you have to do with animals that you guys know better than me, because most of you work on animals, that you associate the odor with a negative outcome, right? Often electric shock. That's the easiest way. Or you make the animal very, very food deprived, right? Or thirsty, right? So you put it in a very bad negative state, right? So, and since it's very difficult to ask the animal how they like its odor, we don't really know how it is experienced, right? But we know in humans, we have a lot of data of both peripheral cortex and orbifrontal cortex has been associated with valence, right? So before we started this project, in the very beginning, we had to develop a way to assess signal from the human olfactory bulb. We basically put different methods together and tweak them to work. So we didn't really invent a new method, we just kind of took existing ones and made them work better together. So historically, when you look at EEG in humans, you cap it at 30 hertz. Anything above 30 hertz, you say is just random shit and you're further away. Historically, the same things goes when you do what's called source reconstruction. We're trying to identify the source within the brain. You normally do what's called skull stripping. So you're removing the bulb from any kind of source that would be of interest. So we basically did, we adopted classical methods to actually fit identifying signal olfactory bulb. And what we found here was a nice signal in the gamma frequency that corresponds to when we, in theory, would think that the olfactory bulb human should start processing the odor. So around starting around 100 milliseconds after odor onset, right? Interesting, while I was doing this, Gary actually pointed me to a chapter in a book from 1958, I thought originally, but I think it's actually 69, because it was published twice, about an olfactometer. And I was building a olfactometer at the time, so you should probably read this one. And I looked at it, and in there, they actually had, when they validated a olfactometer, they actually recorded from a human olfactory bulb during surgery. And that's how they validated the olfactometer work, right? And that's the only known recordings from a human olfactory bulb in a live human, right? And they also found the same activity between 45 to 85 hertz, depending on the odor. So that was a nice demonstration that what we find is likely from the olfactory bulb. The reviewer is forced to do 6 more experiments. And then they rejected us in yes. Then we went into Nature Communication, I think it was published, and we went straight through. But it was a little bit frustrating. But one of the things we did also like source reconstruction, and we can see that was actually coming from the olfactory bulb, the signal source, and this is now an MR image where we are projecting the source onto the brain. You can see that it's in the olfactory bulb region. We also tested test retest, and we have a test retest on 0.80, which is good. We tested people without olfactory bulbs, we found no signals, etc., etc., etc. So we trust that this actually works. So yes.
I always wondered, and you probably know the answer, but you can't actually go into the nose to the olfactory epithelium and put an electrode on the olfactory epithelium. Is it just
my very last slide deals with that. Yeah. So we are developing method, we have developed a method where we do it, but then we record from the olfactory epithelium. But I know that Christina Solano is doing similar stuff, but then she has another type of electrode where she records through the cubiform plate. And she thinks she gets olfactory bulb signal. She's trying to validate it right now. So, yeah. But that's a good question. And now we wanted to know, is the human olfactory bulb tuned to odor valence? And what we did, we recorded this method I just talked about, and I had people come back 3 times because we needed tons of data to do what I wanted to do. And we come back to why all of this data was needed. But just to highlight again is that one of the major advantages we have working with humans, we have a lot of disadvantages. We can't sacrifice our participants. We can't lesion them, etc., etc. We can't do all the things you can do with rodents. But we can do one thing. We can ask them how they perceive the odor for each trial. So after each trial, the participants get the odor and then rate it for perceived valence, right? And then we ignored what odor they smell, and we only look at what they perceived. So all the data I'm going to show you from hereafter is not odor dependent, it's rating dependent, right? Like how they actually perceive it, the valence they perceive it. And the reason why we need to have so much data is that statistically, or mathematically, I should say, you cannot really just compare a rating of a human with a neural response in the brain. People do it all the time. We also do it all the time, I should say. But mathematically, it doesn't make sense because they are in different dimensions. They are in different kind of like space in any kind of way or form you're thinking about it. So instead, we use what's called RSA, representational similarity analysis. And it's basically just a way to put them in the same statistical space so you can directly compare basically any value. So what we're doing, we're building correlation tables. So we had, in this case, we had 6 odors with varying valence. And we just build correlation tables for the olfactory bulb and correlation tables for valence rating and intensity ratings, right? And then we relate when the stimuli in the olfactory bulb, the correlation tables are aligned with what you see in the rating correlation table, right? So now we are comparing normalized values that are in the same dimensional space, right? So we're looking at when the relationship between the stimuli align between the brain and the percept. Everyone follows? So we do this for valence for every time interval while controlling for intensity. And we're using a machine algorithm approach to kind of like compare these multidimensional correlation tables, right? So what we found, well, we looked at what happened in the gamma band first. And here we found, actually, let me walk you through this. So what we're doing is that we're looking at the correlation now between when the brain corresponds with the percept, right? When your 6 odors brain response align with the perception of the 6 odors. And not only that, we're doing this for every individual. We are chunking it in different kind of like time chunks along the 1 second stimuli. And we do it first on the individual level, and only areas that are significantly aligned, we then bring to a second level, and we then do a machine learning approach on that also, right? So it's a very conservative measure. First of all, you need to have a lot of like repetitions to actually create those correlation tables. and only areas of the time frequency aspect that are actually significant will be brought up to the second level. So it's a quite significant way of looking at it. But what we're finding is that finding this early time period around 140 to 200 milliseconds after odor onset, where we have this correspondence between processing in the olfactory bulb gamma band and the valence ratings of the participants. And then we have this a little bit later on, which is sub-significant, but it's definitely something going on there, right? We then turned to the beta band, and we found something reversed. Really nothing going on in the very beginning of the stimuli, but then we had this area here that was significant, right? So after we did this, we found this paper by Leslie Kay, where she showed also this kind of like two-stage model, where she showed that odor processing of olfactory bulb, you have this initial fast processing in the gamma band that is then taken over by more slower top-down informed signal in the beta band, right? And the general theory is that the gamma band is within the area processing, and the beta band is top-down or coming from another area. So more communication going into the region, right? So what we now have is this model, where we have early gamma band processing olfactory bulb, and then we have this two in the later time processing in the gamma and the beta band. What I will not show you, other than the result, we have done a couple of more experiments, but we really map this out. So how is olfactory system communicating odor valence, right? And what we're finding here is that if you look at the coherence spectra between the olfactory bulb and the peripheral cortex, and we try to decode within this coherence spectra, what are they smelling for valence, we see that we can do this in the gamma band. When we go in the other way, we go from the peripheral cortex to the bulb, we can decode what the valence they're rated as in the beta band, and to some extent also in the theta band, right? And then we have other data showing that the peripheral cortex are processing valence in the theta and the beta band, right? So what you can see the general idea here is that the main bulk of the work in the olfactory system in humans seems to be dedicated to the gamma and the beta band, right? What's interesting, this aligned to great extent also with the rodent data, that this is the two main corresponding frequencies. Yes.
How you actually do the experiment. So are you telling the individual that they're about to get a smell?
No.
Something
So this is one of the drawback of the method. If they know when the smell is coming, if you give them a sniff cue, we have this called a CNV response, continuous negative blah, blah. And but basically it's that the EEG kind of like, the whole signal start kind of like being drawn up regulate, right? So we get much lower response. So what we do sniff triggering. So they are asked to sit and breathe through their nose just normally. And then when they are at the very nadir, like at the very bottom of the sniff cycle, then that triggers the photometer. So by the time they start to inhale, the odor will be there. Now, so this is good in a way that the odor is there and we don't get a change in the sniff response in anticipation of the odor. But what is bad, we also know from data, is that when you attend to the stimuli, you get slightly different processing than when it just like appears.
And when did they report?
2 seconds after the odor has ended. So we had like they know every time they're gonna rank or like rate pleasantness, right? So they're aware of it, right? But then we have them wait like this, we don't get the motor response or preparatory motor response, right? That's a good question.
In terms of the time, I see like with the 1 second of multiple dots, and I can see neural-wise you can measure it, but in terms of
No, the dots here just like represents how we chunk the data, because we cannot do this analysis for each second. We don't get enough data, so we're chunking it times like that, right? So those squares just represent, here is the center of the data chunk, you know. But we have a continuous recording, right? So we get millisecond precision.
what about subjective metrics? You were doing correlation in the 2 3 slide people. Are they like also like multiple? Exactly.
Yeah, so they rate every odor. So we have 540 ratings. And we ignore, basically, not in all analysis, but the most analysis, we ignore which odor do they smell. Because we don't care about what's the inherent kind of thing with odor. We care mostly about how did you perceive it, you as an individual, right? Because we all have different experiences. Like, for example, many think the roses smells good. I think they smell horrible, or not horrible, but I really dislike them because I spent 1 year in the basement in Dresden doing olfactory ERPs using phenylethyl alcohol. So for me, rose odor is more of an anxiety triggering signal of being there and seeing this blip, blip on the screen doing EEG, right? So I would rate rose very low, but same thing with vanilla. I think vanilla is like, man, like a 5, right? But maybe you will rate like rose like an 8. So then using the odor rose as a positive, it doesn't work because we think completely different. So we mostly focus on what you are perceiving. Yes, Joe first.
Follow up.
Yeah, follow up, yeah.
The reason for asking about the trigger
Yeah.
is there must have been some time when you just weren't paying attention and they just didn't perceive you better. So how do you incorporate that? And does the absence of perception change the way that the response occurs?
So they had an option to not rate. So we said if you do not, if you don't perceive anything, because even though they pay attention, the weird thing is that sometimes you don't smell the odor. They just like gone, and we don't know why, but this how it is, right? But there are too few trials to look at it. One reviewer asked for that, like, so look at those no odor trials that you get, but it was too few per person. But I think it was like maybe 5 per person. So we need more trials than that.
You can try this on a novel odor to figure out if you can predict valence. You take your model trained on these 6, and then just give them a new odor. You look at the neural signal and figure out.
I mean, these are all, no. Half of them are monomolecular, and half are mixtures. We didn't say difference between the monomolecular mixture per se, but I would say that many of the odors are novel in the beginning.
I mean, one you've never tested before. Brand new odor you've never seen before. Can you predict how the person will rate valence?
You mean like, okay, can we look at the brain response and predict the valence?
It's not part of the training.
We haven't done that specific test, but we have used one set of data from one experiment as a training set and then predicted 2 other experiments. But we asked a slightly different question. But there we can predict odor identity. But we didn't test the valence aspect. But we can definitely train it on we could predict familiarity. So high or low familiarity. If we train on one set, we can in 2 other sets predict whether the odor was familiar or unfamiliar. But we haven't asked that question. Then.
So I missed something. So in the schematic that you showed the electrodes on warhead 4. But is there any location data embedded in this?
So what we do what's called a source reconstruction. So basically, we are interpolating, and that's the main benefit of these 4 electrodes, that we are closing the sphere better. We also try to have electrodes here, but then we get so much motor response. But in theory, we should get better like interpolation from building the whole skull, right? So normally when you have EEG, you stop here with electrodes, meaning that you don't get the source that lies outside, right? So we just like bringing down the source sphere.
And shouldn't they do like that?
We have 4, so they're along the ridge line here. And it's better to have them not just like this and leave it at this, because what we have identified is that the dipoles for the olfactory bulbs, they go like this. They don't go like that, they go like that. So, which is slightly a problem because which means that they little bit overlap. But
just to make the point, you can't extract bad odors high, good odors low from the way that.
No, we cannot go in. We just get one source in the bulb because the source is too big.
Yeah.
We can't dissect it more than just like the bulb, basically.
Yeah, I get you.
And that's why we cannot do the amygdala because it's too close to the prefrontal cortex. So we can choose between amygdala or prefrontal cortex, but we can't have both because then the sources partially overlap, and then we get shared variance, which get parcel out. So. But what we have done is that we can also use this based like fMRI and get sources over the brain. So we can get 2,000 sources and look at how the network are communicating in millisecond temporal positions. So, what you say is that I want to highlight, which I find interesting, and you guys might have a good solution to, is that when we look at this coherence spectrum and we do decoding, we can get it in the gamma band, right? And we also do linear causalities. We show that this has actually the gamma and the beta, right? But now we just decode on the whole coherence spectrum. It's not related to power. It seems to be much more complicated that when it comes to the communication, is something that relates more to the phase amplitude aspect of the signal. So if you have any ideas how we can get to the bottom of that, I would be really happy to hear. So if you look at now this, what's going on in the olfactory bulb again, we can see that this thing here in the early gamma, that seems to be more related to the odorant than the rating itself. And it's mostly carried by the negative aspect of the odorant, right? Whereas this one over here is more related to the subjective valence. So we want to know, is this one here a part of this avoidance system that we want to tap into, right? So what we did, we looked at what's called mu desynchronization, which is like a hallmark in the human brain, at least when it comes to preparatory motor response. So if I tell you to kind of raise your right arm, then just by hearing me saying that, you will have what's called a mu desynchronization between your cognitive thought about it and the preparatory motor cortex, right? So we looked at how the olfactory bulb is linked in this in this gamma band, and we find this mu desynchronization in the preparatory motor cortex that is more for unpleasant than pleasant. So 300 to 400 milliseconds after odor onset, we get this preparatory motor response for negative odors in motor cortex, right? Which I think is interesting, but we'll come back to why I think we can use that. So the model then looks like this. We have a separation in the processing. Some signal go to motor cortex, the other one goes to prefrontal cortex. But if it's true that this is actually an initiation of an avoidance response, 100 to 200 milliseconds after, we should see an actually avoidance response, right? So now we have a prediction we can do. So what we did, we set up a new setup where we measure actual approach avoidance. Normally in human studies, when you measure approach avoidance, most studies are using a joystick. And the theory is that you're pushing way faster on the joystick if it's an avoidance, and you pull it faster towards you if it's an approach. I think it's many steps removed from what you want to look at. So here we actually looked at if you are leaning towards the odor or leaning away from the odor by standing on this sensitive force plate, right? In the first experiment, we didn't know really how should we look at the data. So we had the open hypothesis. So first we looked at around 250 milliseconds after odor onset, because we want to know are they responding to the odor hitting them in the nose, right? This should be around this time, we should have an impact. And then we looked at this time of interest around half a second after odor onset. In other words, this 300 plus 200 milliseconds put together, right? And you can see here is that we have, or I should say also, this is we're measuring now if it's a positive value, the lean forward. If it's a negative value, the lean backwards. So this is the posterior anterior axis, right? But you can see that when you get the unpleasant odor, or odors, I should say multiple, they lean slightly backwards significantly. But when it's a positive, yeah, they move a little bit forward, but it's not significant from baseline, right? And we also assessed, is it more linked to this late gamma beta, but we don't seem to be the case. So we did another study to replicate that, only assessing this time period and pre-registered analysis and everything, and we could replicate that so that they are significantly leaning away from the odor or from the front, then they are leaning forward. All right, and they're not leaning forward. Well, they're leaning forward, but not significant when it's a positive odor. Right? So the critique here that we didn't get during review, but we had it in the lab, is that we also video filmed them, because we have another experiment or project where we're trying to decode valence from facial expression. And we see that some people, they do like this. Oh, you get it? Oh. That means that their center of gravity stays the same, but they're avoiding it. And some people do like this. Oh, that was bad. So the center of gravity stays the same, but the nose is getting away, right? So then we try to see how can we measure true avoidance, right? So we come up with another setup. We're using 3D, it's like stereo photography. First, we tried with laser. We tried 10 different ways of measuring distance, and they all fail miserably. And then we come up with this very simple way of using it's a new type of 3D video where you can get sub-millimeter precision, right? And we have now done a couple experiments using both odors and visual stimuli. And what we're finding here, we can replicate that when it's a negative odor, unpleasant odor, you're increasing the distance between the nose and the odor source. And in the first experiment, they had a nasal cannula, so they couldn't really get away, right? The odor followed them wherever they went, right? Now we are blowing odors towards them, so they can actually get away from the odor. The problem, though, is that they're getting away from almost every odor because there's an odor coming towards them. So we also have an avoidance response for the neutral and the pleasant, but more so for the unpleasant, right? So we can show that we have an overt behavioral response to negative odors, where initially it seems to be tuned towards the inherent information contained within the odor, right? And then the later seem to be guided more by information from the full olfactory system, coming down, beaming down from the peripheral cortex, where of course you have this input from past experiences, labeling, expectation, and the like, right? And I just want to take a short detour here and show that some other data we have. Let's see if you can see this. I can do it like this, make it more, where we measured what is happening in peripheral cortex when we are seeing and hearing stuff. So we did one experiment. We put 50 people in the MR camera, and we showed them visual and auditory stimuli of objects. Never in the whole experiment are we mentioning odors at all. Right? They believe they're just lying there trying to identify pictures and sounds, right? And while we're doing that, we see that particularly here in the particularly here in the posterior peripheral cortex, we have screw it. It's better to zoom in. Particularly here in the posterior peripheral cortex, you see that we have significant activation both of visual stimuli and auditory stimuli. And what's interesting here is that the posterior peripheral cortex is thought to be the part of the human brain that start formulating the odor object, right? After the experiment, when they're outside from the camera, we have them view all the visual stimuli and listen to all the sounds again. And for each stimuli, rate the degree of odor association to the stimuli. Because they could be, even though we don't have any odor information in the camera, they might subconsciously or somehow thinking about, oh, this smells a little bit like this, right? And that will provoke this kind of activation. But we're finding no such things, that even some are very strongly odor associated, some are not. We don't find any relevance, right? And where am I going to go with this? Well, where I'm going to go with this, the peripheral cortex is a very multisensory area. So already here, we have help to build up the olfactory perception from both the visual and auditory system, which makes sense, right? Because we are inherently really bad in identifying odors. So, and we very seldom outside the lab are experiencing odor in isolation. I would say that 99.9% of all odors that we are exposed to, we have some clue or hint of where the odor is coming from and what is actually smelling. What we also have found in further studies that we have what's called phase amplitude coupling between the beta and the gamma band here in this late time aspect of the bulb processing, which we think corresponds to an updating of the olfactory bulb priors, right? So you have this information coming from peripheral cortex about like this odor you just experienced, past memories, associations, and that is then meant to update the gamma, which in turn will update the gamma processing for your next sniff. Because historically, we always look at the first sniff, right? The first exposure. But in real life, this is very seldom we go into a room, it's like odor, and then walk out, right? Most of the time, we go into a room, oh, it smells bad. Oh, it smells really bad. We take multiple sniffs, right? So the system needs updating constantly. So we then did a very simple experiment. We recorded brain response during 2 sniffs of the same odor. So we keep the odor on now for a couple of seconds, and we see what's happening for the first sniff and the second sniff of the participant. And our question is very simple. How is a human olfactory system in olfactory bulb updating the information? How is that mechanism happening, right? And our theory is that the gamma or the beta activity in the first sniff will predict the gamma activity in the second sniff, right? Because we know we can decode from the beta activity what they are smelling, right? We're not as good in gamma, but definitely in the beta we get rich information, right? So this is very preliminary data. My student analyzed this for he sent me this for like a week ago, so he's still like working with the data. But what we're seeing here is this. I see he didn't put in time, but this represents around 4.5 seconds, right? So the odor goes on here, and this is now the normalized sniff curve of the participants. You see that here we have this initial gamma, and then we have around here is a beta. It just looks a bit because we have massive gamma going on later here, which means that we are tuning down all the representation of the image, right? So, but what we see is that if we use a signal in the early beta, in this late beta, around 700 millisecond, 500 to 800 millisecond after the onset, we can actually predict the gamma activity in the subsequent sniff in a very precise manner, right? If we do the opposite, if we take the activity in the late gamma, which I thought we could use, we cannot predict what's going on in the second gamma. And I think the simple explanation to why the gamma prediction fails is that we get enormous amount of gamma in the second sniff. So if we now contrast the what's more in second sniff versus the first sniff, you can see that throughout the whole period, we get much more gamma activity. And we get a very strong reduction in beta activity during the second sniff. Yes?
I don't know how to look at this graph. What is the beta? What is the gamma? How can you see it?
Oh, sorry. Yeah, I should tell you. So this is now the frequency. And this is the time, right? So it's basically a time frequency spectrum of what's going on in effect about. We make one for the first sniff and one for the second sniff. And then we just basically do a t-test between the two. So what we're looking at here are t-values, so statistically significant difference between the two. And this is what is represented by more in the second sniff. So you're not looking at the raw underlying power, you're looking at statistical differences between the two representations, right?
When you say that beta predicts gamma, where is the beta? Where is the gamma?
Yeah, so the beta is here. This is the first, you don't see it so well because we're scaling it to this one, all right, which is much higher. If we zoom in on here and scale it according to this one, you will see that you have some beta activity here. And then we're trying to predict what's going on. Oh, sorry, yeah, I should have gone over oscillations. Yeah, sorry. Yeah, when we're talking about oscillations, you historically mostly focus on alpha, which is the lower frequencies of the band, right? And then when you go a little bit higher in the frequency, we talk about the beta band, which is around 12 to 30 hertz, right? Okay, yeah. And then the higher is the gamma frequency, right? And historically, when it comes to human EEG, we have been capping at 30 hertz because we thought that anything above 30 hertz, nothing to pay attention to. So if you look at the old literature when it comes to human olfaction, they talk a lot about alpha and beta because that's what they could see because they didn't include the gamma. But what we can see is that if we go beyond the first sniff, we get massive amount of gamma activity, right? Which, and I talked to Dima Reinberg 2 days ago, and he basically finding the same thing. They're doing the same thing now in animals. What's going on in the second and third and those, and also finding massive amount of gamma for some reason. Right? And we're not sure what it means, right? But I think the take-home message from this one, though, ignore the frequencies and all the things I'm talking about. The take-home message for me as a psychologist and behavioral interest is that already in the second sniff, have you extremely strong cognitive modulation, which means that in olfaction, and this is the olfactory bulb, right? In olfaction, I don't think you can separate perception from cognition. Because if you already in the very first synapse has a very strong cognitive modulation, then what is perception? Right. To conclude, odor valence is linked to avoidance behavior and is at the first sniff at least, mostly guided by odorant inherent value, but thereafter heavily influenced by top-down processes, right? And it's a dynamic connectivity that goes over a range of frequencies, depending on direction, change over time. And I think the take-home message to this extent is that even though human and animal models show large similarities in the result between what we are finding and what's in the literature for the animal models, there are systematic differences for humans and animals that would be interesting to explore why is it so different, right? And they're not linked to the sniffing frequency. There are other aspects there at play that we don't know yet, right? So where are we going with this, right? So now we're presenting beta and gamma and theta and gamma, and let's just say like, what are the stuff, right? And that's a valid question, like why do you know what is communicating and how, right? That's because what are we doing now in the lab? First of all, we have a setup now where we can record from LFPs from the olfactory epithelium in humans. At the same time, we can record from the olfactory bulb and the peripheral cortex. So for the first time, we can basically follow the signal from the olfactory receptors to the brain and back. And our main interest here is not so much like how is the signal going upwards, but my personal interest here is trying to understand why is 70% roughly of the connections between the bulb and peripheral cortex going downwards. Why do we have so much top-down projection into the bulb, whereas in all the other sensor systems, it's the reverse, right? It's 70% upwards into the brain. So we're focusing heavily right now on understanding this top-down modulation all the way down to the olfactory receptor neurons, right? We also have two new systems in the lab we are setting up now. One is called focus ultrasound, another one is transcranial temporal inference stimulation. And basically, there are two different methods that can do the same thing. We can change the neural activity in an area. So with the focus ultrasound, we can downregulate cortical structures on any depth because we have crossing beams, basically, right? So the beams, each individual beams, they go through tissue without changing the neural activity. But where they meet, we create an envelope, and that will then downregulate the neural activity. But so that's one thing to kind of like the shut down areas. We can explore then what are they contributing in the system. So get some kind of like validation of, we can really we can modify the system, right? But more interesting, this one, it came out 2 years ago. It's called transcranial temporal interference stimulation. And basically what that is that you put electrodes on the scalp, and then you have high frequency electric signals going down into the brain, right? And since they're so high frequency, they're between 2,000 to 5,000 hertz, they don't really change the neural activity or anything, right? But where they meet, you can create envelopes. So you can actually create 20 hertz or 30 hertz or whatever you want in the bulb or the pineal cortex. And since you now know how the processing are happening and where, we can now start modifying this. So we can change this early gamma activity to go slightly higher or slightly lower. Because if we dig into the data, we get different frequencies depending on the odorant, but also like the individual percept, right? So we hope we can actually go in and modify activity in the bulb and pineal cortex, and then change the percept of the individual. And why is that of interest? Well, if we can start actually creating percepts or changing percepts, we are the first step when it comes to neurostimulation, when it comes to implant in patients, right? And that's why we're doing all this mapping to understand how is our olfactory system communication. Because when we know the neural code, actually beaming in the electricity in the brain is very simple. We do it all the time in tons of different patients, right? That was all I had. Thank you very much.
Go back to your first thing, and I'm not clear as to whether you think that there are inherently negative odors.
I think there are a few inherently negative. They might not create a negative percept in all individuals. But I think that the first time you're exposed to them, I think they are inherently negative. One inherently negative is so I'm from the very northern part of Sweden where I was born. And we have something that's called rotten herring, which is viewed as a delicatessen. You harvest them 1 year and you eat them 1 year later, and it's fermented fish, right? And my wife, when she had it the first time, she actually vomited because it's a horrible odor, right? But once you get used to eating this, you actually start to liking it, right? But the initial exposure, I think, is negative for everyone who feels it the very first time.
And there's a set of receptors that are targeted for negative odors, you believe?
No, I wouldn't go so long to call it like a label line. No.
So the other thing you said right at the beginning was people have a special thing that is really good because they can talk. But quite a bit of what you showed, you don't need talking, particularly the movement to and fro or whatever. And I wonder whether you have are interested in looking at very young children with those kinds of measures, which, as far as I know, have not been done with odor.
So one of the things that is good, this new approach avoidance method we have, is that people are not aware of what we're doing. We don't tell them in measuring how you respond to the odor. And in the first study, when they were standing on a force plate, so they know that we're measuring how they're moving, right? But now we just have them stand in the room, there's nothing on them, and we tell them that we just want to know how you like these odors. So after each odor, tell us a number from 0 to 10, how you like it. For each picture, just tell them how much you like it. So they are completely unaware that we are assessing the approach avoidance, right? Which is a good thing. We have tried similar stuff with infants. So we have gone to the delivery ward, and we took the force plate and we put infants in little cradle. We looked at odors, and we are finding some response that they are kind of like at least getting like leaning a little bit away. And this is 2 days, 1 day or 2 days old infants away from the negative odor. We don't find anything towards positive or their mother's odor though. Limited sample. We're looking for money to do a bigger study on it, but that could be one way one can look at actually this kind of like more mode response. Right?
I mean, I certainly agree with you that negative is much more important and much more salient than positive, and that's true throughout all of psychology.
Yeah, but I would expect them to lean more towards the mother's odor. but we simply cannot find that.
It's hard to measure this, like facial expressions. It's not so clear, but negatives are much, much clearer.
Yeah, it could also be that they're not so good in doing the motoric with the whole body. Because we have Ben Rashal's data, and I think maybe you guys also have it, that they are like looking a little bit towards the mother's odor, right? In the incubator.
Maybe.
Yeah, but we're finding so much variance in the motor response. So the negative is the only one we find that they're really twisting the whole body away.
So Joel and I are interested in different levels in the idea that there are sort of classic odors that make up all of the odor world, and you can mix them right to make. And I think maybe that's a little bit why he was asking a question about a novel odor, which I'm interested in as well, because certainly what we found and we were older children was that the children's inherent responses that we investigated were very, very much aligned with what adults did. And so, I certainly think that there are these inherently negative odors. But it's difficult to prove, of course.
It's difficult, yeah. I mean, we also involved, we got this quite substantial money from European Union to digitalize olfaction with Monell Spring. So we're also going after like what is the basic odors that from which you can create all other odors, right? And I think there are probably some core odors that are most important, because if you look at the olfactory receptors, there's only a few receptors that are the workhorses, right? And then you have all the other ones that just like complement the picture, basically.
So at the very end, you sort of threw up this piriform LFP slide, but you didn't tell us anything about technically. Is that you're in patients or in healthy subjects to get LFPs?
Healthy.
And can you tell me more about how you're getting?
It's basically
it's an electrode.
Yeah. So it's source extraction. And I have some slides on that actually. Now people online will not see this, I assume. Where we are assessing, can we really get signal from the piriform cortex? And the two dots here, left and right piriform cortex, and then all dots you see are all other sources, right? Alternative sources. And we get a reliable signal from left and right piriform cortex, right? But we have to go through to quite an extent and length to get a signal. We need to get like an MR image of each individual's own brain, right? And then we have to have what's called neural navigation, which is like a $100,000 system to get the 3D space of all the electrodes. And then we have to have some kind of like computation. And if we go through all that, we can get a good source, right? So we think, or what we know, we're getting signal from the olfactory epithelium, the bowel, and piriform cortex reliably. Yep.
I have two questions. So with the opium signals, so if someone lost the sense of smell, you see it.
So if someone do not have a sense of smell, can we see? Yeah. So we have done testing on people with isolated congenital anosmia. So they never smell an odor. In many cases, they don't even have an olfactory bulb, and we don't get any signal in those. It's just like from the bulb. From the piriform cortex, we get signal, right? And we have used both EEG and fMRI. And what we see is that we're publishing the fMRI data now is that piriform cortex in smell-blind individuals are overtaken by visual and auditory stimuli to a varying degree. And if you're going to get a cochlear implant, what they often do, they make you lie in camera and see visual stimuli. And if you have visual processing in auditory cortex, you're not a viable candidate for a cochlear implant. Because once a cortical structure has started doing other stuff, it's really hard to make it go back to what it did originally, right? So I think that it goes to show that if you lose a smell for a long time, you have to go in the intervention early, because the olfactory cortex is a prime reality space that will be taken over by the visual auditory system.
So my second question, with the gamma and beta, so how, I wonder if you can, are they correlated to anything we know from animals, like what kind of neuron response or?
So you're asking what generates the gamma and the beta frequency, right? That's a good question. That is a little bit discussion, right? But the underlying what's causing is spiking, right? Which is interesting because that's actually something we can look at when it comes to scalp electrodes. We can look at what's called burst activity, which is a correlate of spiking. And that's something we're now digging into. Can we look at, in this case, beta burst in the prefrontal cortex and the olfactory bulb, which will then be an analogy to spiking. But it's basically the underlying thing is the oscillations of spiking neurons, right? That are spiking different frequencies.
It sounds like when you're recording these waves, you're actually kind of getting information of the whole signal from that brain region.
Yeah.
And you're losing the individual cells and what they're saying.
Yeah, definitely.
At the very end, you mentioned that you have this very cool thing where you could government maybe interfere with this whole wave. But I'm just wondering what's happening at the cellular level because you might be creating a waveform that replicates what you see, but by activating random cells in a random order, and so it might not actually lead to the outcome that you want.
Yeah, and I think that's a more of a problem in olfactory bulb. Piriform to some extent, but the bulb, I definitely think we can have a problem because piriform is the olfactory bulb is so depending on inhibitory mechanisms. So if we go in there and create like let's say a 60 hertz frequency in the bulb, what are we triggering both the activating area but also the inhibitor area or one or the other? We don't know, right? Because it's such a new technique, no one really looked at it. The other problem we have is that the signal will travel. So even though we stimulate area A, if area A is connected to B and C, area B and C will get some of it also. Because one area drives the other areas, right? And what that is doing, we don't know, and we don't know really how to address it yet, right? I mean, we're just starting using the method, and it's been right now out for a year only. So, but it's definitely we have hundreds of experiments I can think of where we have just controlled stuff, right? So.